Hello and welcome everyone. My name is Leah Sabin and I'm an executive director in the Regeneron Genetic Medicine Department. And this is the Nucleus by Regeneron, a podcast where we talk about exciting science and geek out about the particular areas of science that we care about. Our guest today is Janice Chen. She is a co-founder and chief technology officer at Mammoth Biosciences. We're really excited to have you for the podcast and welcome.
Thanks for having me.
Sure. So to kick things off, I would love to dig into your backstory and find out, how did you get into science? What brought you to, pursuing a scientific career, and really how did you get into the CRISPR field in particular?
Yeah, so I guess I can go back to my childhood. So I grew up in Salt Lake City, first-gen Chinese American, and grew up in Salt Lake City because my dad was doing his PhD at the University of Utah in pharmaceutical chemistry. So at a very young age, I was exposed to the lab. I have a lot of fond memories visiting my dad in the lab, playing first with the consumables, like the Eppendorf tubes. And so I always have associated the lab as a fun place to be. I think, that exposure really helped me build a natural affinity to science, but in particular, biology and chemistry. So I guess fast forwarding to undergrad, so the other part of like how I got really interested in the specific field of CRISPR kind of starts when I was fortunate to meet Dr. Jeff Buca, who was a professor at the time at Johns Hopkins, and he had a course called Build a Genome. And, it was just an amazing vision for me because the idea was to build the minimum yeast genome from scratch.
As an undergrad.
So again, this was like a huge team, but as the undergrads in the lab, we were the ones like running the PCRs, stitching these DNA oligos one by one to create larger chunks, which would become chromosomes. So it was just really exciting project, which involved like bioinformatics, genetics, molecular biology. So
that's amazing.
Yeah. So that's what eventually brought me to PhD. At the time I was still deciding what to do after undergrad, but I decided that, I wanted to really go deep on the science. And so I did a year at Harvard Medical School with Pam Silver, who's one of the leaders in synthetic biology. So got to just work on some really exciting projects, reprogramming bacteria to produce biofuels and all kinds of crazy stuff, right? So that was, again, super exciting, but also I realized that the tools that we had available at the time, actually, CRISPR was just like coming onto the scene, and we were still doing old school, genetic engineering, if you will. And it was very clear, like there was a lot of opportunity to improve the processes that we had. So that was when I made the decision. I was like, okay, I'm going to go to grad school. And I was lucky enough to join Jennifer's lab. And that's where I also met Lucas Harrington, who is also a co-founder of Mammoth. And that was sort of the beginning of where we are now in the world of CRISPR.
Well, so we're going to be talking about CRISPR a ton today. So to level set everyone on what it is, why we care, could you, I guess, explain in simplest terms, what is CRISPR? Why is it such a big deal? Why is everyone talking about it? And what advances did it open up for science, for medicine, for, everything that we do?
Yeah, so CRISPR stands for clustered, regularly interspaced, short palindromic repeats. You don't have to remember that, and I always get it wrong. But what's really fascinating about CRISPR is that it is a natural bacterial immune system. So in the same way that you and I have an immune system to defend against viruses and bacteria, bacteria also have a natural immune system to defend themselves against invading viruses. But what's most important for why we care about it is that one part of that CRISPR biology is what we call a nuclease, and that is a protein that actually is able to target that invading virus and just chop it up. So what scientists had done, this was back in 2012, Jennifer Doudna, Emmanuel Charpentier, and Martin Yannick and others in the lab, they were able to extract that core CRISPR protein that was doing all the cutting of the nucleic acids, and they were able to show that you could reprogram it to target any sequence. And that was the beauty of CRISPR, is that all of a sudden this idea that you could target anywhere in the genome, but you could do it in a programmable fashion, and that really frankly changed the world, changed research as we know it. And
right. Well, and I want to go back to that era when you were really first joining her lab. But before we get there, how generally is it thought, I guess maybe for the CRISPR 1.0, so like the basic nuclease, how can that be used therapeutically, and why are we so excited about that piece of it?
So I mean, biology is like amazingly complex, but there's also our understanding of the genetics behind certain diseases has become more sophisticated over time. And so there are certain disorders and diseases that have a known genetic basis, and oftentimes this may be because you have expression of a faulty protein, which just creates, some disease phenotype, or, there's a particular gene that's either upstream of another, process in a pathway that either is faulty or is creating either upregulation or downregulation of a pathway that you don't want, and shows up in a disease phenotype. So the fact that we can just cut a gene with CRISPR 1.0 is just think of it as like totally just knocking out the activity of that protein. So when the protein is faulty and you don't want that disease protein around, if you just cut it out, that can result in a knockout of that protein, and you can rescue that disease phenotype and get to a healthy state for the cell or for the person.
So the original discovery, at least in Jennifer Doudna's realm, had happened before you joined the lab. Yeah. So at the point when you joined, what was the focus of the lab, and really what led you to you and Lucas together to co-found Mammoth, and walk me through how that happened and what the basis of that was?
Yeah, gosh. When I started the lab, it was like the Wild West of CRISPR, because nobody, there were just so many open questions about the Cas9 protein, about the biology of CRISPR, about, what else was out there. For me in particular, I was always really excited about the fundamental mechanism of the protein. So I was looking at it from a very mechanistic standpoint, but it was super exciting because this is like an amazing molecule that has a protein component and RNA component comes together, right? And then it just does all these functions. And trying to understand sort of that underlying mechanism, it was so exciting for me. I wasn't even thinking about the application at that point, right?
Right. So you're really you're really an engineer tool builder.
Exactly. Yeah, sort of. Exactly. Yeah. And actually, Lucas and I, we sat next to each other in the lab, so we were literally sharing a bay. And so he and I would constantly just like throw ideas at each other, criticize our ideas, right? It was a very productive and collaborative relationship. And I think we really pushed each other too to really think, what we could do with these technologies. And because of that, evolutionary history of these CRISPR systems, we also realized that, there was so much more to the CRISPR universe. And so a big part of the theme in our grad school career was really trying to chart new frontiers in the CRISPR space, so looking at new enzymes, understanding how they work, because that was a critical step to then apply them to specific applications. We didn't really realize it at the time, but looking back, it was, again, we had all these foundational technologies, and I'm sure we'll talk about, what those were, but specifically within these like ultra compact enzymes, which was a totally new paradigm for the field, because people thought that, it's a small protein, like it's just an artifact, like it's not going to work. But we actually showed that it worked and what that could enable. But all of these pieces of the puzzle that we were working on, we ended up, in-licensing from Berkeley, and that became the foundation of Mammoth. And we realized there was really no better opportunity to really push this vision of transforming our discoveries into, ways to transform patient lives. There was really no better opportunity than to just do it ourselves. And so we, paired it with Jennifer and Trevor, and this was 2018, and we launched Mammoth as sort of this next-gen CRISPR company.
Okay, so let's talk about Mammoth today. Could you tell me about the major focus of the company and really where your niche is in the broader CRISPR field?
Yeah, so our goal is to develop in vivo gene editing therapies. But, to sort of put it into context, I think it's also helpful to describe sort of how the field has evolved over time.
Absolutely.
You mentioned CRISPR 1.0, and that was the genesis of a lot of great companies as well, right? Using Cas9 for single gene knockout has a ton of applications. But I think as that space has evolved, it was clear that, the applications were not limitless, right? It wasn't like the one size fits all. And then CRISPR 2.0 sort of happened in that interim. This is probably while for the most part, Lucas and I were still in grad school, but this is where you saw like the toolbox really expanding. So going into, more precise gene editing modalities, changing single bases, writing small sequences, modifying, the epigenetic editing. And so this was really, looking at diverse ways of creating different types of editing outcomes.
You're right. And I feel like that's the evolution of the field, which, in the beginning, it was a bit, I guess, archaic at this point, which seems ridiculous because we are only about a decade in.
Yes.
However, it's moving from sort of a blunt double-strand break towards these more sophisticated editing modalities. So could you sort of give a little bit more color on why the field is moving in that direction?
Yeah, I mean, so you talked about the blunt sort of double-stranded breaks. I think if you create a double-stranded break in a genome, like the cell has to figure out how to repair it because that in itself is a toxic event. And so the outcomes of repairing that double-stranded break can result into unintended consequences. And I think that's been sort of one of the motivations for the field to say, okay, hold on, like we have a way to program this CRISPR protein to go somewhere, but can we do something besides just cutting it, right? So that was kind of the motivation where people were like, hold on, like now you can think about CRISPR as like that scaffold and let's just decorate it with different types of, proteins that can do other things.
And so what are some of the things that Mammoth in particular, what kinds of other bits and bobs are you attaching to your CRISPR nucleases, or I guess dead nucleases, to give them different functions?
Yeah, so maybe before I talk about what we attach to it, we can talk about the core enzyme itself, because that really is kind of the engine that really makes Mammoth special. And I think also why, we are so excited about working with Regeneron as well. And that really comes down to these really small CRISPR enzymes I think I alluded to earlier, but they're just about a third or half the size of your traditional gene editors like Cas9, which means you can actually add all these exciting functionalities to that system that is now also compatible with delivery modalities. So we always think about the ultra-compact nuclease as like the core scaffold. And if you have the capacity, and we create that capacity by working with these small enzymes, you can start to add on things like, base editors, like reverse transcriptase, to write in small sequences. So the sky is kind of the limit in terms of what you append to the scaffold. And again, that CRISPR piece just becomes like the GPS system to wherever you want to go in the genome.
Yeah. Well, and I think the ultra compact size of your nucleases was a major driving force in us starting to work together. But the reason that we were so excited about working with Mammoth is because Regeneron's really been focused on that delivery piece. We've been trying to use antibodies to make delivery of genetic medicines more efficient. But one of the huge limitations with one of our favorite delivery vehicles, AAV, is its packaging size. And so in looking for a partner, we were really looking for the ability to deliver not only the basic nuclease, but some of these enhanced functionalities that that Mammoth has been developing, but still maintain that small size that would be compatible with our delivery.
Exactly. And we're not a delivery company. We do fundamental innovation on the CRISPR side. And so seeing the advances you've made and really rethinking the delivery problem, for us, it was so exciting because, as you pointed out, that therapy has two components to it, right? And so this really kind of brings the best of both worlds.
I agree. Well, and I think that it's even showing up at the meeting that we're attending right now. So, we are in this conference room, but in reality, we are also in New Orleans, and we're here attending I'm bad with acronyms, American Society of Gene and Cell Therapy, or ASGCT. Yes. I've been sort of monitoring the themes that are emerging from the different sessions, and the meeting is far from over. But are you excited about any given theme or talk, or what have you seen so far that has excited you or that you're looking forward to hearing about?
Yeah, well, I'm biased because I am the chair of the ASGCT Gene Editing Committee. So we actually organized the gene editing session, which the theme was all about advances in large insertions, right? So that session happened this morning. So, I listened to all those talks with great interest. And I think what was really also surprising, well, not surprising, but cool to see is kind of like this theme of like what's old is new, right? All of a sudden we're talking about like recombinases, like
absolutely.
These things have been around for decades. And then, zinc finger nuclease is kind of like the OG gene editor. So people are really being creative about, let's take the learnings over the decades, in the space, and let's apply it to really creative ways. But then there's also new innovations like the bridge recombinases. So, totally new things that have been discovered in the last year as well.
I agree that really editing doesn't always have to involve CRISPR per se. One of the exciting advances that I saw in the presidential symposium is the idea of using epigenetic repression to suppress prion protein expression in the central nervous system. So using evolved delivery methods, next-gen delivery methods, and next-gen payloads together is really where I think things are going, and it's exciting to see it progressing.
Yeah, and that's where Regeneron Mammoth, I think, are really pushing the boundaries too, and super excited to see where these go.
Well, you had mentioned earlier that a lot of the initial attempts, or I guess the initial ways that we can envision employing editing is often in the liver. So it's sort of, the first place to start. And in my mind, that's because it is the most accessible organ through a systemic delivery route with a number of different delivery vehicles, lipid nanoparticles, AAVs, a lot of them go to the liver. Yeah. So I think a key challenge in the field has been getting outside the liver. Could you comment on why it's important, in particular with CRISPR systems, or I guess editing systems in general, why not just focusing on the liver is of interest and a motivating factor for even why we are working together?
Yeah, no, I totally agree that liver is a very tractable problem, but it only will address a subset of the totality of genetic disease. So I think, we always think about how can we make the greatest impact with the technologies that we have. And if you only can go after one organ, you are just going to miss out on changing, and curing diseases, the vast majority of diseases, in fact, right? And so we must think about ways to solve this problem. And I think that's the beauty of like looking at it from different angles, because some people might say, hey, I'll just like shave off pieces of the Cas9 protein and get something that's good enough. But then you're gonna hit a wall again, right? Because we talked about genome surgery and all the decorations you got to put on the protein, and now you run out of space, right? So if we just fundamentally start with something that just gives you the capacity to be creative in how you create that editing outcome specific for the use case that is compatible with a delivery modality that can go, hopefully anywhere, then you really have something that's going to create that kind of sea change of impact.
I totally agree. Now, while we're developing these types of therapies, one very common question I'm sure you get a lot is, what are the pitfalls or the downsides of using editing, and how do we navigate off-target effects?
Yeah, I spent a lot of time in grad school thinking about the off-target problem, because that was a big motivator for me, is like, what are the mechanistic reasons why you would see an off-target cut versus not? And actually, in some cases, the rules are well defined. In other cases, it's a lot more complicated. But the short answer is that only is part of the problem, because at least in the early days of looking at off targets, we were completely ignoring the delivery stuff. Like, it was just, if we threw a Cas9 into cells, like, what do you see, which is first important step. But like, when you think about in the totality of like a human or a patient, then you also have to layer on the target, the delivery specificity aspect too, right? The biodistribution.
100%. Yeah, I know. I often draw a quadrant, and so it's on target editing in the intended locus,
yes,
on target editing in an off target tissue, off target editing in an on target tissue, and then off target editing in an unintended tissue. And so, how do you develop the assays you need to look for all of those, and how do you do that when really you're looking at a human genome? So how do you model that in a monkey or a mouse? And all of those questions are 100%. Really challenging.
And I think you talked about like the multidimensionality of the problem too, which I think is something that the field is still figuring out how to do. And actually, related to a talk at ASGCT, right? And you brought up like, how do we build the assays to look at it? Because I think the field also realized some of the assays that we had early on, you were just you were just looking at what you're looking for, right? But there's all this other stuff that could be happening, like these large, kilobase deletions that we were totally missing because we just weren't looking for them, or we weren't looking for it in the right way.
Right.
And I think that's an important learning for all of us in the space, which is like you have to think about how do you design like an unbiased assay to capture which are these huge, genomic rearrangements that you could totally miss if you're not looking for it.
Yes. So I think it really speaks to the need to continue on the quest of new technology development and not potential, even aside from engineering the nuclease, it's, engineering all of these assays, next-gen sequencing-based methods and beyond to figure out the effect that your drug is having on a genome and where and when.
Exactly. Yeah, it's a totally new type of medicine, so new ways of looking at that. Yeah.
Well, a lot of Regeneron's delivery efforts historically have focused on AAVs or adeno-associated viruses, but we definitely have an interest in extending those observations to other types of delivery modalities, and one of them is lipid nanoparticles. And I think the reason for that interest is very much in the editing world. The beauty of a lipid nanoparticle-based delivery is its transient nature. So you can have exposure to your nucleus and then, it goes away. So what are your thoughts there? I mean, do you agree?
Really big, part of the puzzle. And the way that we always thought about it is that we wanted our technologies to be compatible with any kind of delivery system. We see the transient property of LNPs as a highly attractive. The ability to deliver, mRNAs within that lipid nanoparticle context also, you sort of have fewer variables in some cases that you have to worry about, right? That you would in an AAV context, like you said, with like the capsid and that hanging around in the tissue.
For sure. One additional challenge that I think about a lot is immunogenicity. Where do you see the key challenges continuing to lie in the CRISPR field, and how should we be focusing our innovation as we continue to work together and as the field at large continues to move forward?
Yeah, I know the immunogenicity problem is interesting because I remember in the early days, people were like, oh, the original Cas9 comes from a human pathogen, and so are there, pre-existing antibodies, and it was a big point of concern. And then I think it sort of empirically was not a big issue, right? So that was kind of interesting. That being said, I think the immunogenicity, certainly on the AV side, I think that keeps me up at night, honestly, right? You can't redose, right? So you have one shot.
Yeah.
And know better than I do.
On that note, though, we actually have a presentation at this meeting about multiple programs that we are pursuing to try to address that exact problem.
Cool. Okay.
So as you know, Regeneron pursues genetic medicines, but we also have a really rich antibody portfolio. And what we're trying to do now is look into our toolbox and a different toolbox of antibodies this time and see which of those can actually be used in immunomodulation, and can we use existing Regeneron assets that might change the way the immune system responds to certain stimuli, and can we use those in the context of genetic medicine? So we're working on trying to solve the problem of redosing, to be able to suppress immune responses, whether they be B cells or T cell responses to that initial dose of AAV, to try to prevent those responses from forming and allow redosing. We also have a program that seeks to deal with the other big immunogenicity problem, which is pre-existing antibodies to AAV. And so the idea is hopefully we can make advances in both these areas that would allow more patients to be eligible for trials and allow patients to potentially get a second or third dose, and maybe even intra-patient dose escalation if we're successful.
It's super exciting to hear, because I also think about, there's, like you said, there are patients that are on AAV therapies but maybe are not efficacious. And so if they cannot, get a future AAV therapy, maybe AAV CRISPR therapy, that's hugely problematic, right? So
absolutely. And yes, and we heard in the presidential symposium some advancements in the use of a compact nuclease to try to perform exon skipping in the case of Duchenne muscular dystrophy. But I think as we transition that initial approach to humans, there invariably are going to be efficiencies that just aren't there from mouse to human. So to have the ability to for those first patients that likely will receive a suboptimal dose, for them to receive a second dose in the future would be a game changer.
Exactly. I totally agree.
So this has been a really great conversation, and I've really enjoyed hearing about your backstory and also talking science. So thank you for joining us, Janice. It was really a pleasure. Our guest has been Janice Chen, co-founder and chief technology officer at Mammoth Biosciences. And I'm Leah Sabin, and this has been The Nucleus by Regeneron. Thank you.
Thanks, Leah.